Scientists may have identified a hidden molecular kill switch behind Alzheimer's for decades. Alzheimer's disease has been studied primarily through one dominant lens: the buildup of amyloid plaques in the brain. But a growing body of research suggests that amyloid might not be the root cause. It could be just one piece of a much larger puzzle. Now, scientists have uncovered a far more direct mechanism that may actually be responsible for killing brain cells, a kind of molecular kill switch embedded within neural signaling systems. Researchers led by neurobiologist Hilmar Bading at Heidelberg University have identified a specific interaction between two proteins that appears to actively trigger neuronal death. This discovery shifts the focus from passive damage like plaque accumulation to an active destructive signaling process within brain cells themselves. If you like my videos, subscribe my channel and press the bell icon to turn notifications on, so you keep getting all the major advancements in science and technology. And don't forget to like and share this video with your friends and colleagues. A dangerous twist in normal brain signaling. At the center of this mechanism are two well-known biological components: the NMDA receptor and the TRPM4 ion channel. Under normal conditions, NMDA receptors play a critical role in brain function. They sit on the surface of neurons and respond to glutamate, a neurotransmitter essential for learning, memory, and communication between brain cells. When these receptors operate within synapses, the junctions where neurons connect, they actually promote cell survival and strengthen neural networks. But location matters. Outside synapses, NMDA receptors behave very differently. When they physically interact with TRPM4, they form a previously underappreciated complex. And this is where things go wrong. This pairing transforms a normally beneficial signaling system into something harmful. Instead of supporting neural activity, the combined structure begins triggering stress pathways inside the cell, ultimately leading to degeneration and death. Researchers describe this structure as a death complex, not as a metaphor, but as a functional switch that pushes neurons toward destruction. Breaking the death complex. The most striking part of the discovery isn't just identifying the problem, it's that scientists may already have a way to interrupt. The research team tested a specialized compound called FP882, designed to target the exact interface where NMDA receptors and TRPM4 bind together. This region, referred to as the TWINF interface, acts like a docking site that allows the toxic complex to form. FP802 essentially blocks that connection. Instead of shutting down the NMDA receptor entirely, which would be dangerous, since it's vital for brain function, the compound selectively prevents it from entering its harmful configuration. In other words, it doesn't silence the system, it stops it from turning toxic. What happened in Alzheimer's models? When tested in mouse models of Alzheimer's disease, the results were surprisingly strong for an early-stage intervention. Animals treated with the compound showed significantly reduced neurodegeneration. Synaptic structures. The connections between neurons were preserved. Mitochondria, which supply energy to cells, remained more functional. And perhaps most importantly, cognitive performance improved, with treated mice maintaining better learning and memory abilities compared to untreated ones. Even more intriguing, the treatment also led to a reduction in beta amyloid accumulation. This is a critical detail. It suggests that the toxic signaling pathway may not just be a consequence of Alzheimer's. It could be feeding back into the disease process itself, accelerating plaque formation. If that's true, then interrupting this pathway could have a dual effect, protecting neurons while also slowing one of the disease's most visible hallmarks. A shift away from the amyloid-centric model. For years, most Alzheimer's therapies have focused on removing amyloid plaques or preventing them from forming. While some of these approaches have shown modest benefits, they have not delivered the kind of breakthrough many hoped for. This new research represents a different strategy. Instead of targeting the visible symptom of the disease, it focuses on a downstream mechanism, the cellular machinery that actually executes neuronal death. In doing so, it reframes Alzheimer's not just as a disease of accumulation, but as a failure of cellular regulation. From a systems biology perspective, this is a major conceptual shift. It implies that neurodegeneration might be driven by misregulated signaling networks where normal biological components become harmful under certain conditions. That opens the door to more precise interventions that restore balance rather than simply removing debris. Broader implications beyond Alzheimer's. What makes this discovery even more compelling is that the same NMDA-TRPM4 interaction has been implicated in other neurodegenerative diseases, including LSTY. That raises an important possibility. This death switch may not be unique to Alzheimer's. It could represent a shared vulnerability across multiple brain disorders. If that's the case, then targeting this mechanism could lead to a new class of neuroprotective therapies, ones that work across diseases rather than treating each condition in isolation. The reality check. Despite the excitement, it's important to stay grounded. These results come from preclinical models, specifically mice. Translating such findings into human treatments is notoriously difficult. The brain is vastly more complex, and many compounds that work in animal models fail in clinical trials due to safety, delivery challenges, or unexpected side effects. There's also the question of timing. Neurodegenerative diseases develop over years or decades. Intervening at the right stage before irreversible damage occurs will be critical. So while FP802 and similar compounds are promising, they are still far from becoming approved therapies. A new direction in brain research. Even with those limitations, the significance of this work is hard to ignore. It demonstrates that we are beginning to understand neurodegeneration, not just as a buildup of harmful substances, but as a dynamic process driven by specific molecular interactions. And importantly, those interactions can be targeted. In the long run, this could lead to treatments that don't just slow Alzheimer's, but fundamentally alter its trajectory. Because if scientists can truly control the switch that tells brain cells to die, they may finally be able to stop the disease